Hello, and welcome to this video on protein interactions. At the end of this video, you will be able to explain how proteins interact using non-covalent bonds and define and explain allostery, probably the single most important topic we're going to talk about all year. So proteins can act as individuals, but they will often bind together to regulate each other or to create new functions from their complexes. In order to do that, they typically use non-covalent or flexible interactions. So for example, in this picture, you've got a protein shown in green, and you've got a ligand shown in yellow, which may or may not actually be a protein, but for the purpose of this conversation, let's assume that it is. And when they fit together like this, you can see that they have so many non-covalent interactions holding them together. Remember that non-covalent interactions are less strong than covalent interactions and therefore more flexible. That allows these molecules to come together with what's called binding affinity. Binding affinity is essentially a measure of how strong an interaction is. In fact, binding affinity is a measure of how quickly things fall apart, but the quicker something falls apart, the less strong its interactions were to begin with. So when we look at this in the space fill model, what you can see is that these things are close to each other, but not actually touching. So if I just kind of draw a circle around that yellow molecule, but they're interacting through ionic, hydrophobic, and hydrogen bonds that hold these pieces together. And remember that the individual pieces, this green protein right here, is built with covalent bonds. But these interactions between the molecules are probably non-covalent. And remember that that can be ionic, hydrogen, or hydrophobic interactions. So when these proteins bind together, the number and types of interactions determine the strength and the duration of that interaction, which again can be defined as the binding affinity. So the protein that makes this up has several layers of structure. Remember that the tertiary structure comes from the side chains. So why don't all molecules fit together equally well? Well, each protein is made up of different combinations of amino acids that give them different potential interactions they can have with other molecules. So here you have a protein shown in green, and it's only showing some of the amino acids, but it's showing their side chains in colors. So you see that the serines and the threonine are there in yellow. Those are polar amino acids. You've got your glutamic acid, which is negatively charged, and your arginine, which is positively charged. And they're forming different interactions because of the chemistry of those individual side chains. The hydroxyl group here on the serine has a partial positive charge that can interact with the partial negative charge coming off of the phosphate of that cyclic AMP in the middle of the picture. This arginine has a positively charged amine group, which can interact in an ionic interaction with the negatively charged phosphate coming from that cyclic AMP. Glutamic acid is making hydrogen bonds over here. And the serine is again making hydrogen bonds now between this amine coming from the cyclic AMP and the hydrogen coming from the serine. So the more of these interactions you have and the stronger those interactions are, the more time these two molecules will spend together versus apart. And so molecules are always coming together and falling apart, coming together and falling apart. And binding affinity really asks, yes, but how much time did they spend together? And how much time did they spend apart? If they spend a lot of time together, we'll call that a strong interaction with high binding affinity. Some proteins have multiple binding sites, which allow for regulation. So for example, enzymes often have an active site where the substrate fits in, but then an allosteric site for regulation. So in this picture, you see that the active site is over here. The substrate will come in, and then it will break apart into products. You've got two active sites here. But each of those also has a regulatory site on the side that can bind to this molecule, CTP. And when it's bound to CTP, the active site itself has changed shape. It's changed structure such that the substrates can't fit in there anymore. So let's think through that. When the molecule is bound to CTP, then it changes its shape in response to that interaction that it's having with CTP, and now it can no longer bind to substrates and turn them into products. Essentially, we turned the enzyme off simply by letting it bind to CTP. This is the most important concept of this semester, and it's called allostery. When two molecules interact, their shapes change slightly to form interactions with that new molecule. This is often known as induced fit, or as I call it, the bum in an easy chair molecule. If you've ever sat in an incredibly comfortable, very squishy chair, you know that as you sit down into it, the chair itself adjusts its shape to conform to you. And in fact, you kind of get a little bit flabby and lean in and change your own conformation to really settle into that chair. Likewise, molecules will do this when they bind to other molecules. Their shape changes to accommodate that new molecule and to improve the interactions between them. I'll show you what I mean. In this picture, you see hemoglobin. Hemoglobin carries oxygen. And so everything on the outside in these pinkish, purplish colors is all hemoglobin. And on the inside, you see this bluish colored molecule drop in to the active site. That's oxygen. So what you see is this induced fit. When the oxygen dips in there, the whole molecule changes its conformation just a little bit to accommodate that new interaction. And if the oxygen leaves, it reverts back to its original conformation. All molecules are doing this all the time. Every time a molecule interacts and binds with another molecule, it changes its shape just slightly. This process is called allostery. Molecules have multiple stable conformations, which are determined by their binding partners. So it's not just that molecules have multiple stable conformations, it's that which conformation they're in depends on who they are binding to at any given time. The more binding partners a protein has, the more possible stable conformations it likely has as well. Some of those conformations might be functional, and we call them on. And some of them are maybe not functional, like the enzyme we saw earlier, and those are off. Regardless, the point is that we have ways of regulating how proteins function by changing who their binding partners are and using that to make them more or less active at the jobs that they do. We will see examples of allostery nearly every day in this class. For example, here you see a G protein coupled receptor, and the G protein is shown here in green. When the orange receptor binds to this G protein, you see that the G protein changes its shape in two ways. First of all, it tucks itself up underneath that receptor. So you see that that light blue protein has a new part that's poking up into the receptor. But in doing that, it also created an opening here where GTP is going to bind. So that light blue and that kind of dark blue protein used to be closed up tight, almost like jaws that are closed. But as soon as it binds to that orange receptor, allostery occurs, and that opens up the protein so that it can accept a new molecule, in this case, GTP. Again, the most important concept of this semester is going to be the idea of allostery. And so in order to understand that, when we're looking at specific examples of allostery, we should always be asking ourselves, what interaction led to this change? And what is the consequence of that change in conformation? What's the consequence of this interaction? Again, we'll see this every day, so we'll get lots of practice identifying and understanding allostery.